Hi there, my name is Andrew Eckford, and I will be your instructor for this part of the JTG IEEE Summer School in Information Theory, Signal Processing, Telecommunication, and Networking. And this part of the school is called Introduction to Molecular Communication. So as I said, I'm your instructor, Professor Andrew Eckford from the Department of Electrical Engineering and Computer Science at the Lassonde School of Engineering in York University, Toronto, Canada. So to get started, a little bit about me. I've been at York University since 2006 as a professor, and I've been working in this field, molecular communication, since about 2007. Way back when it was brand new and there were maybe 5 people in the world working on it. So I've been around since the very beginning, and this is my work, this is my expertise. So you're about to benefit from everything I know. If you want to get a little more of a sense of my research, you can visit this link. This goes to my lab website, eckfordlab.org. And you can get a better sense of the things that I'm working on right now, which is both classical molecular communication and some more recent applications to biological systems, including some neat applications we've found to things like plants, to microbiology, to some fairly cool applications elsewhere. And I'm going to show you another thing here. Well, I'm not going to click on it, but I'm going to leave the link here. This is a TEDx talk that I did. Many people find this, and it's an extremely high-level introduction to the kind of work that I do and what I think about it. So a lot of people like to see this as the first thing that they hear about me, and I welcome you to check it out. It's not technical at all. It's the sort of thing that would appeal to a very wide audience, but you might want to check it out if you wish. So what are the goals of this course? I have the following 4 goals. So there is an outline found on the course webpage that states the basic things that we're going to learn. But my goals for this course are to give you a good, but not too heavy introduction to the mathematical and physical basics, to show you the way towards information theoretic results with a couple of examples, to survey the state of the art in molecular communication, and to give you a couple of examples of practical systems and inexpensive lab work. Starting with the first point, to give you a good but not too heavy introduction to the mathematical and physical basics. This is potentially the most intimidating part of molecular communication, because as communications engineers, or people who are generally interested in communications, we normally have an electrical engineering background. And if you're anything like me, when I first started thinking about molecular communication, I had not taken a course in biology or chemistry in decades. I didn't really remember many of the most important principles. Well, I'm here to tell you that what you need to know in the mathematical and physical basics of molecular communication is not very complex. So it is definitely different. It is significantly different from things that you would have been exposed to in electrical engineering, but it is not all that difficult, at least as far as the basics are concerned. And what we're going to do in this course is we're going to introduce you to those basics, just the simplest things that you need to understand in order to get started. We're going to move towards communication and information theory, and we're going to show you how to communicate using chemical principles, using molecules, and we're going to actually start right in this lecture giving you the very simplest ideas of exactly how to communicate using molecules. We're going to survey the state of the art in molecular communication. Again, with those basics that we talked about, we're going to discover the range of work that's being done right now in research, both in terms of molecular communication itself, and in wider applications, for example, in biology. We're also going to give you a way to get started right away. So again, another intimidating thing about molecular communication is that you might think that you need very expensive laboratory equipment. You might need expertise that you might not have. I'm going to show you the systems that we have built without a wet lab, without complicated equipment that costs maybe a couple of $100, a couple $100 US in order to get started. And it's just very, very simple hack together things that I think anyone could do. So it's very easy to get started. The message that I want to give you here is that it's easy to get started. It is not difficult to pick up the basics. And we're going to cover, it's not difficult to make research contributions. And we're going to cover exactly how to do that in this course. By the end of this course, you should hopefully have enough background to at least start reading the literature, if not making contributions yourself. Again, this is an introduction, and I want to give you an idea of how I'm going to approach this course. So a lot of the material that we're going to talk about is mathematical. So I'm going to use two methods. When we are talking about high-level concepts, as we are talking about right now, I might use slides. So there are going to be some slides such as this, and I will provide those slides for download if you like. But when presenting mathematical material, my preferred way of presenting it is to write on the blackboard. So there's two reasons for that. And I find that this works very well with technical material. When I write, it forces me to slow down. So when I write notes, if I write mathematical scientific notes, it forces me to slow down and make sure that I'm expressing every concept clearly. When slides are used for that, I find that the temptation is to go too fast. So that's why I like to do it. There's another reason for this, which is, as I'm going to get to in a second. Well, first, let me just say, when presenting this mathematical material, you'll see how it works. I'll write notes. I have a tablet. I will connect the tablet and record the screen. And I will write notes directly to that tablet and share the screen. And I will record those notes and I will share them later, but I strongly recommend that you write those notes in your own notebook as I write them on the tablet. So for example, if I write an equation on the tablet, what I recommend that you do is that you find some way of getting a notebook, be it your own tablet or something else, and that you actually physically write those notes in the same way that I write them. And that's the second reason why I like to do this on the blackboard. Not only does it slow me down, but it slows you down. It forces you to write every concept that I am explaining. And there's something in the process of writing it down, I find, that really helps students process everything that is being said in a way that does not come across on slides. So that's why I'm only going to use slides sometimes. I'm going to use it for conceptual things that are not terribly technical, like this introduction, for example. But a lot of my lecture will be me writing on the tablet as though I were writing on a blackboard in front of you. And again, I strongly recommend that you get your own notebook somehow and that you write notes as I write them. I think this will help you to understand. As far as the structure of the course, there will be 8 hours total of lecturing. And what I plan to do is I plan to divide those 8 hours into 30-minute or so chunks. So it's not one huge block. Of course, over the course of this pandemic, we have had a lot of experience with online delivery of education. And I find that this method of dividing course material into approximately 30-minute chunks works pretty well. So that's how I'm going to do it. So in other words, there's going to be probably 16 chunks of this lecture. They'll be divided up by topic, and I will provide those to the organizers for posting. In fact, you probably found them now that you're watching me right now. Okay, the structure of the course. These are the topics that we're going to basically cover again. This is taken from the course webpage, but how it's going to work, we're going to start with the introduction, and you are here. So this is the introduction. We are going to review the physics and mathematics of molecular communication. So again, those are those concepts that I talked about a moment ago. The new physics of chemistry, of how molecules propagate, of how to express information in molecules, and the mathematics of how that might work. So we're going to go into some detail, but again, I don't want you to feel intimidated by that. The physics that we're going to use are relatively easy to understand and relatively easy to model. It's just very different from any kind of electromagnetic physics that you've probably encountered in your work so far. We're then going to delve into the communication theory of molecular communication. So once we've thought about the physics of how molecular communication might work, we're gonna look into exactly how communication theory can be applied. And it turns out that communication theory is sufficiently general that any physical principle can be adapted. It doesn't necessarily have to be electromagnetic. From there, we're going to move on to some advanced concepts that are the subjects of more recent research. We're going to talk about mobile molecular communication and multiple input, multiple output, or MIMO molecular communication. We're going to talk about the information theory of molecular communication. So that's pretty interesting. We're going to talk about the ultimate limits of what these systems can do. And I think you're going to be surprised at their capabilities. It turns out that you can actually communicate quite a bit of information using molecules. We're then going to talk about biological communication. So biology is a recurring theme in this course. Many of the applications are biological. It takes its inspiration from biology. So we are going to talk about biology throughout this course, but in this section, we're going to talk specifically about how to use molecular communication to analyze biological systems, and in particular, signal transduction, which is how microorganisms communicate with each other, and the cells in your body communicate with each other. And finally, as I mentioned, I'm going to show you some experimental approaches to molecular communication. I'm going to give you some examples of devices that we use in the lab that are very inexpensive, that don't require a lot of expertise, that are basically just hacked together components that you find off the shelf, or that you can order online for very cheap. You don't need a big complicated lab for this. Let me also say that I am, roughly speaking, lecturing from this book. Full disclosure, I'm a co-author of this book. We published it several years ago. We're actually in the process of updating it. But if you want more information, or if you want a book to cross-reference what I'm saying, if you need a little more information, if you like to read out of a book while a course is happening, you can get this one. And I'm going to try to make my lectures correspond as much as possible to the material in this book. Okay. So for the rest of this lecture, again, this is an introductory lecture. So we're gonna start very easy. We're gonna introduce the very basics of molecular communication. So what is the most basic thing we can talk about? Well, we can talk a little bit about how to express a bit. Like, how do we actually communicate one bit or a few bits of information using molecules? We're then going to delve into a little bit of the history of where this field came from. And you're going to find that it has actually kind of a long history, dating back at least 50 years, although most of the communication work has happened within the last 10 to 15 years. And we're going to talk about, why do we care about molecular communication? What's important about it? We're going to talk, broadly speaking, about the applications to biotechnology and biology. There will be more details on applications far later in this course, but I do just want to touch on it briefly as part of this introductory lecture. Okay, so the very basics of molecular communication. What are the simplest things that we need to know about molecular communication? Well, if we think about communication, we should normally think about this model. This is the block model that they teach in communications courses. So I would assume that you are familiar with this. If we're transmitting digital information, we start with a set of messages that we could possibly send. I'm going to call it M. We pick one of those messages. I'm going to call it little m. So these possible messages are enumerated. There's a finite number of them up to the size of the sets. We pick one of those, we take one of these, and we call it little m. We pass it to a transmitter. The transmitter figures out a way to encode our message across whatever medium we're using to transmit the information. So that's called modulation. In the medium, the message is affected by noise. So for one thing, the message must be transformed in some way to be expressed properly in the medium. And it is affected by distortion or noise or some mechanism by which the message that we send is not necessarily the message that we receive. So at the receiver side, we observe the message that was sent, M, in the presence of the noise, and we try to make a guess. And we guess, the receiver will guess that the message was M prime. And the goal here is to ensure that when we guess M prime, that it's equal to M. And if we don't do that, then we make an error. So what I just described is the very simplest model of communication that is taught in every communication course. And what's interesting about this is that I didn't say anything about the physical mechanism by which the medium operates, or the transmitter or the receiver. As long as I can pick a message and encode it somehow, the medium is abstract. So we know that in conventional communication systems, the transmitter is a modulator and antenna. The medium is free space. The noise is additive white Gaussian. The receiver is a receive antenna and an amplifier. Actually, the noise occurs in the amplifier at the receiver, but we can consider that part of the channel. And then we are very familiar with ways of guessing what the message is at the output of the additive white Gaussian noise channel. All we have to do now is transform all of these blocks into mechanisms that will work with chemistry, with molecules. So in other words, we need a chemical transmitter. We need to think about the chemical medium. We need to think about chemical noise. And we need to think about how a chemical receiver would work. So again, this is extreme basics. We're going to go into much greater mathematical detail later. But I just want to give you a quick idea of how this might work. How would you say it with molecules? How would you encode a message, a binary message, such as this? So here we have a binary vector. I've chosen it randomly, and I've just displayed it here. So the idea is we want to figure out a way to express this message using molecules. So how might we do this? The first thing to notice is that if you want to transmit bits, in other words, you want to transmit ones and zeros, what you have to do is you have to have two possibilities. This is very obvious. So each time you send a bit, you have to send a 1 or a 0, and that means what you need to do is you need to make a choice. There must be something that you can use to make a choice. Now in communications, in electromagnetic communications, we know how to do this. What we do is we have, for example, in phase shift keying, we have one signal that's with 0 phase, another signal that's with 180-degree phase. With frequency shift keying, we have 1 or 2 frequencies. For example, to send a 0, we have frequency F0. To have a 1, we have frequency F1. So all we need to do is we need to think of ways in which to make a choice in chemistry. And there are really 3 basic ways to do it. So one way is identity. We're going to talk a lot more about this later in the lecture, but some people call this method molecule shift keying. So in other words, we're going to have a little reservoir at my transmitter. By the way, cell 1 here is the transmitter, cell 2 is the receiver. So cell 1 might have 2 reservoirs, and in order to send a 0, it might release molecules from one reservoir. Let's call it A. Maybe these are sodium atoms, sodium ions. If I'm going to send a 1, then I have another reservoir of type B. Let's call these calcium ions. So in other words, now I can make a choice between one of two chemical species. This is making the choice in identity. If I want to send a 0, I send sodium or type A. If I want to send a 1, I send calcium or type B. And then The receiver's job is to measure the identity of those arrivals, and then it can detect exactly what was sent. So in this example, it measures a type B or calcium molecule, and so therefore it realizes, oh, it must have been a 1 that was sent. And then we can use that same principle to encode a vector by sending sequentially type A and type B, depending on exactly what bits we want to transmit. On the other hand, we could signal using quantity. In other words, if we want to send a 0, we could release nothing. So my transmitter, by sending a 0, decides to release nothing. And if I want to send a 1, I could release a certain number of molecules, let's say 5 in this example. So in other words, if I send a 0, I send nothing. If I send a 1, I send 5 molecules. I could just send 1 molecule. You could send a million. It doesn't matter. But the key difference here is that you make a choice between 0 and 1. And then the receiver's job in that case is to measure the number arriving. Now you'll notice that we released, in this example, we released 5 and 2 arrived. So that's interesting. That actually gives us a suggestion of how noise might work. So what might happen is that when we release these 5 molecules, for one thing, propagation isn't instantaneous. I mean, it's not instantaneous in electromagnetics either, but worse than that, it's a lot slower in molecular communication, and moreover, it's random. So what could happen is that the other 3 molecules might follow strange trajectories. They might completely miss the cell. They might follow very convoluted trajectories. So they might still be in the middle here, but they might have been delayed by going back and forth with a random walk. So at the decision time, I might only observe 2. And in fact, I might observe 0. They could have been that they all got lost, in which case I will confuse the 0 and the 1 at the receiver. But nonetheless, I can make a decision here. I can make a choice here between sending a 0 and 1 in the quantity of the molecules that I release. Finally, timing. So this is a little bit different. So what I'm going to do is, if I want to send a 0 or a 1, I can send a 0 by releasing a molecule immediately. So we can assume that the transmitter and receiver are synchronized. Never mind how, we can talk about that later, but let's assume they're synchronized. They agree on certain times during which symbols will be sent. So if I can send a 0, if I want to send a 0, I can choose to release a molecule now. If I want to send a 1, I wait a little bit. So I wait a little bit, and then I send it at some release time t greater than 0, still within that same symbol interval, but I wait until later. So this is a little bit like pulse position modulation that we would be accustomed to. At the receiver, what we do is we measure the arrival time. So if it arrives early, it's probably a 0, and if it arrives late, it's probably a 1. But again, molecules can get lost. The propagation time is random. So perhaps we sent it early, but it was delayed by a long time through the random walk, and it arrives late, and we confuse it for a 1. So there's noise here, clearly, and it should be clear that the source of noise is consistent. It's the random walk from transmitter to receiver. But the point is that I can make a choice. I can choose a 0 or a 1 by adjusting the transmission time of the molecule, and that's detectable. So we've got 3 ways, and these are the 3 basic ways to communicate with molecules. They are adjusting the type of molecule, the number of molecules, or the time of release of molecules. There is actually another way. What you might have noticed is that we're always using basically the same molecule. Okay, that's not quite true. At first, we talked about adjusting the type, but you can generalize adjusting the type to the point where actually you have so many different kinds of molecules that you can actually start to inscribe information on these molecules. So some people have talked about emitting custom DNA strands that actually contain your entire message and letting them propagate. So I guess that would be an extreme kind of identity of communicating using molecules in terms of their identity. But these are the fundamental types of saying it with molecules, and there are other ways, but they're not widely used. These are the ones that you see the most. Okay, so now we know a little bit about how you might communicate using molecules. I think it's fair to pause for a second and think a little bit about where this idea came from. So why would we bother? What's the basis for this idea? And it goes back. I mean, it goes back a long time. But what people normally point to is a famous lecture that was given by the physicist Richard Feynman in 1959, and it was called "There's Plenty of Room at the Bottom." In Feynman's lecture, he indicated that so this was, of course, in the 1960s, and he indicated that there are very few physical barriers to getting smaller, and he indicated that there's many ways to shrink physical objects, and he in fact showed that, or he showed through simple arguments that things like microscale or even nanoscale machines were perfectly feasible. A lot of people give Feynman credit through this talk for inventing more or less the field of MEMS or NEMS. So MEMS is micro electromechanical systems. NEMS is nano electromechanical systems. Other people think that these ideas were around and Feynman simply popularized them. But nonetheless, starting from about 1960, people began to give serious thought to significant miniaturization well beyond anything that had been thought of before, to the point of actual nanoscale, microscale or nanoscale devices. On the communication side, of course, we're all familiar with Claude Shannon's famous paper, A Mathematical Theory of Communication. Others had, at around the same time, at Bell Labs, started to formulate the mathematical theories of communication. And what's interesting is that around the same time, actually a little later, but not too much later, like in the late 50s and early 60s, it was found that there were immediate biological applications for this work. One of the first was a paper by Johnson and Knudsen, which actually applied information theoretic analysis to kidney function. And more or less what they were doing is they were showing that decisions are made by the kidney. So basically what the kidney does is it filters toxins from the blood. It allows toxins to pass through it and into the urine, and keeps the blood where it is. So they formulated this in terms of a communications and decision problem that actually applied information theory. Another early leader in this field was Toby Berger, who introduced rate distortion theory, and he indicated much the same thing. And in fact, there was a role for communication in terms of the functioning of biological systems through minimizing the distortion. There's also a neat model that was introduced in the 60s. It was called Blackball's chemical channel. And it wasn't really introduced as a way of thinking about chemistry, but others came along and realized that in fact it does seem to have a little bit to do with chemical communication. So because it's kind of a neat example, I want to go into it a little bit. So basically what Blackball said is imagine a bag. Okay, so this is just an image I found on the internet of balls. So imagine a bag filled with balls like this. So there in advance, you know that, let's say you know that there's a certain number of white balls and a certain number of black balls. The transmitter is allowed to introduce one ball, either white or black, into the bag. So that will clearly change the relative proportion, whether you choose white or black, that will clearly change the relative proportion of balls. And at the receiver, you're allowed to pull one ball out, either white or black. So Blackball said, this is clearly a communication channel. The input is affecting the output. The input and output are dependent on each other. And what is the capacity? So it turns out that that is still an open question. People haven't completely answered it. There's ways to answer it in special cases, but the general case remains open, which is surprising for such a system that's simple to describe. But it's also clear that this is chemical in some way. If you imagine what's in the bag here as a chemical system. So let's say the white molecules are water and the black molecules are salt or something like that. Then by introducing water or salt, we actually change the concentration, and then we can detect that changed concentration at the other side. So this has been thought about as potentially a way of analyzing chemical systems, and we take a little bit of inspiration from it in molecular communication. More specifically, as far as molecular communication as a term is concerned, the term was coined, in my view, by this 2005 paper by Satoshi Hayama and several other co-authors, where the paper was actually titled Molecular Communication. So this is the first time, I think, that anyone came out and called a communication system that used chemistry molecular communication. So as we've seen, there's a long background of this, but 2005 was the first time, I think, that anyone came out and called it that. Like I said, at the time, there were maybe 5 to 10 people in the world working on this. I happen to be one of them. But since then, in the last 15 years, there have been over 1,000 papers published. My textbook, Molecular Communication, was published. There have been 1 or 2 other textbooks as well. There's a new IEEE journal, the IEEE Transactions on Molecular, Biological, and Multiscale Communications. There have been IEEE and ACM conferences. And there's even an IEEE Communication Society Technical Committee. So as you can see, the field is growing, it's thriving. There's tons of research to do, and we're doing a lot. It's an exciting time, and it's an exciting field to be joining. So you might wonder, well, why are we doing this? Well, what are all of these people doing? So very, very briefly, I just want to talk a little bit about the applications that people think of in terms of biotechnology and biology. Starting with applications to biotechnology. So this is really the science fiction-y type of application that we think about. We imagine small, tiny robots the size of blood cells that navigate through the body and perform medical tasks. So molecular communication is the enabling technology for what we call nano networks. The issue is not at all that these devices are overly futuristic. So in fact, these devices, tiny devices of this kind, are old news. Richard Feynman thought of them 50 years ago, 60 years ago, in fact. And even over the past decade or longer, people have been able to build them. Here are some images that indicate some devices, some nanoscale or microscale devices that have been actually produced in labs. So tiny robots such as this. This is an American 10-cent piece. Small devices that can be implanted in individual cells. These are electron micrographs of particular machines that are basically of the size of cells. This particular one right here in the lower middle, what it does, basically these little blocks can connect, and they can make simple structures by connecting to each other. So these devices well, not easily necessarily, but they can be built, can do this. We can actually get those devices into the bloodstream. That's not the hard part. The hard part is getting them to do something interesting. So getting them to cooperate in order to perform a complex task is difficult. So cell-sized robots can be built, but they must collaborate to perform complex tasks. And molecular communication was devised as a solution to the problem of how to do that. So it should be clear that radio actually doesn't work very well for this problem. If you're inside the body is well approximated by salt water. It's very difficult to transmit an electromagnetic signal through that environment. It's difficult to create the hardware at this scale. Antennas aren't very efficient at this size. But molecular communication is the solution that is actually used in nature to communicate amongst cell-sized devices. Bacteria, for example, use quorum sensing. The cells in your body use signal transduction. So molecular communication is biologically inspired. It's intended to solve this problem, the nano networking problem of connecting very tiny devices. So looking forward, if we can solve that problem, what can we do? We can do drug delivery. So we can imagine devices that can seek out tumors and deliver chemotherapy drugs directly to those tumors, increasing the toxicity to the tumor and reducing the toxicity to the patient. Or we can imagine these robots can proceed to the site of an injury and reconstruct the injury, maybe even constructing something as large as an entire limb. We can imagine all kinds of things. We can imagine nanosurgery. These devices could fix a knee ligament, and then once done, they can flush themselves out, so that no complicated surgical intervention would be needed. Really, the sky's the limit as far as applications go if we can solve this nano-networking problem. And that is why molecular communication is very interesting. So the applications are at the intersection of MEMS, NEMS, that's microelectromechanical and nano-electromechanical systems, and biology. And I hope I've made clear that communications are central to these kinds of problems. At the same time, there are non-nanoscale applications. We'll talk a little bit about these probably later in this course. But before we get to the nanoscale therapeutics, which are probably decades away still, there are still reasons to study molecular communication, because we can actually design systems like this that solve problems that we have today that are not well suited to radio. This is a robot we built in my lab, and one thing that's interesting about chemicals that is not very well suited for radio is that chemicals are persistent. So what you can do is you can lay down a little tag. So in this experiment, what we do is we put little alcohol drops spaced out uniformly, and then we take this robot and drive them over the tag, and we see if we can read the tag. And in fact, we can. So this, in this plot, we see the output of the alcohol sensor with respect to time, and we can see that as we drive over the chemical tag, it reads it. So there's 1 spot, 2 spots, 3 spots, 4 spots. And this has applications towards things like heavy urban search and rescue, navigation underground, navigation in the deep sea, anywhere where radio doesn't work, or even just where you need to leave a tag to indicate something to a swarm of devices. So there are clearly both nanoscale and non-nanoscale applications. There are clearly futuristic ideas. There are clearly near future ideas. This is a profoundly exciting area of research, and one that I'm very glad that you've expressed interest in. So to conclude this introduction, and which is to be continued throughout the rest of this course, this is a field with new physics. It's a new field. It's rapidly growing. It has a long history, but there's tons of people and tons of research in being involved. There are exciting futuristic applications as well as practical within reach applications. So let's get started.